Assalamualaikum warahmatullahi wabarakatuh. Salam Namo Buddhaya. Good morning to all of participants that join here in our meeting room and join via online. And also to our honorable speaker, my peace be upon all of us.
It is an honor to us to introduce and welcome Dr. Paul Hutchinson, PhD, from National University of Singapore. For a brief moment, we would like to invite you to stand up and sing the Indonesian national anthem, Indonesia Raya.
This guest lecture is part of our attempt in implementing the three main purposes of the university, which is the first one is to increase our knowledge in the field of flow cytometry, especially related to infectious disease, such as maybe tuberculosis, because tuberculosis still endemic in Indonesia, including in Bali.
In our teaching hospital, we have flow cytometry, but until now, we only use the instrument for leukemic phenotype. And for next time, we hope we will use flow cytometry for the other case, like infectious disease.
Then, on behalf of Faculty of Medicine and University, I would like to express our high appreciation and warm regard to you, because you already share your time, come to our faculty, come to our university, that we need a lot of help of you, of course, in the future, especially.
We are now continuing our meeting to the main agenda. That is to hear the lecture that will be delivered by Dr. Paul Hutchinson, PhD from National University of Singapore.
Thank you for the invite. It's great. We've had a great time in Bali. I think the workshop went very well. I think a few of you attended that, and I'm sure we'll do a few more in the future as well. So I'm going to give a talk today, which I did this at the workshop, but I'll probably take a little bit longer today because I think we've got a bit more time. Talking about flow cytometry in infectious diseases. So just let me move that up there. So I don't know what your immunology background is, but let's sort of go back to the basics. The immune system response against infection, it can be split into two groups, the innate and the adaptive arms. And the innate system consists of cells like neutrophils, macrophages, soluble factors like complement, etc. They can quickly respond to non-specific signals from any infectious organisms which might infect the body. The adaptive immune response develops after this initial innate response, and it's directed specifically against the organism causing the infection. So it's a targeted response. The two main cell types in the adaptive immune system are the B cells that make the antibodies and the T cells that drive cellular immunity. And it's about the T cells is what I'm going to talk about today, and how we can measure their function in infectious diseases. So two main groups of T cells, the CD4 and the CD8. CD4 T cells, known as helper cells, because their main role is that of helping or directing other immune cell responses to infection or other threats in the body. They release cytokines that can also enhance the innate immune response, so factors like interferon gamma. And talking about cytokines, so as I said, one of the things T cells do when they're stimulated by their antigen is to make cytokines. So they'll rapidly produce and secrete cytokines after that antigenic stimulation. To measure the cytokines by flow cytometry, we need to trap the cytokine within the cell so we can stain it with antibody. If it releases the cytokine, it's too late, we've missed it. But if you can block that cytokine release, and you can do that with Golgi transport inhibitors, Monensin or Briffeldin, you can then trap those cytokines in the cell, and then you can do intracellular cytokine staining. So you normally do a surface staining first to identify, is it a CD4 or CD8 T cell? What are its memory markers? And then you do a fix and perm to do the intracellular staining of the cytokines. And it's known as intracellular cytokine staining, or ICS. So this is the results from our study. So it's back in 2005. We looked at 3 groups of patients at the Monash Medical Centre in Melbourne. We looked at adults with newly diagnosed active TB as evidenced by culture positive, or in the absence of that, high likelihood based on clinical, epidemiological and investigational data, and that they were going to get anti-TB therapy. So there was 15 in that group. Then we looked at Australian-born or from other low-incidence countries and who had a diagnosis of pneumonia but had an alternative microbiological diagnosis or a very low probability of TB. And then we got some normals, healthy controls from the lab. So blood was taken, we did that 6-hour stimulus, and then we did the intracellular cytokine staining assay. So this is looking at the response to PPD. Okay, so I'll finish there. Flow cytometry is an ideal technology for analysing the role of T cells in infection. It can quantitate and characterise the function of pathogen-specific T cells during infection, and these measurements are likely to have near-future clinical applications in infectious disease diagnosis and treatment. And thank you for that, and happy to take questions.
What is the sensitivity and specificity of flow cytometry in detecting the presence of Mycobacterium tuberculosis? And then, is the cytometry can displace the TCM or rapid test molecular like a gen expert? Thank you, Dr. Paul.
I would say it's not up there with the gene expert yet. So this gives you an indication of the sensitivity and specificity. We don't have it listed there, but the area under the curve, that was the best that we got. I don't know the exact values, but I think it's as good as this and getting better. How it compares to gene expert, there's been a few studies comparing the two, and I think normally the flow is getting to that sort of range. I think my understanding, the problem with the gene expert is it doesn't always give you a positive response when there's actual infection, and particularly in TB uveitis, I know that they don't get a very good response there. So I think we should look at all different assays, what can work, and definitely PCR-based assays are very sensitive in that, you would hope. So at the moment, they would probably be more sensitive and specific than ours.
Okay, thank you, Paul. Can you show the result of research from Prof. Jusak,
Yeah.
because you said that the cross-sectional study, so the patient before and after therapy is a different patient or
Yes.
one?
Different patient. Yeah.
Okay, different patient.
Yeah.
So,
it would have been nice to see if they all went down, but no, it's just cross-sectional. And it was 8 weeks after treatment.
yeah, okay.
Yeah.
So we don't have the data for the patient after treatment that they have before treatment.
No.
So yeah, different patient.
Yes. Yeah. We only had a 2-week window to collect the patient. So it wasn't ideal, but we were able to get some information.
Okay. Pictures, this flow cytometry for infections is only for research. You think like that?
I think it still needs research. We still need to do research. I'm hopeful we could get some sort of clinical test from it. You know, it's like the quantiferon assay in a way. I mean, the quantiferon is you take this blood, you stimulate a day, and then you measure the cytokines. Well, the only thing we're doing different is we're staining the cells. Whole blood assays are a lot easier to standardize than isolated cells. So, you know, the quantiferon, they've developed it very well. You take the blood direct into the tube, stick it in the incubator, and then assay the supernate.